Okay, here we are. This is the second half. I'm going to share our screen. There it is. And we're going to do our, here's your little PowerPoint. Yay! All right. I'm trying to and now it's muting when I'm not telling it to mute. Oh my goodness. And I guess it see, keeps on muting on me. Oh, technology. You gotta love it or hate it, I guess. Okay. All right. So, second stage of respiration, the Krebs cycle. Let me move me out of the way. Oh, I don't want to make me bigger. I want to move me out of the way. There we go. And we're going to start with that. You know, I keep on saying three-ish. So here's the ish part of it. It's called the intermediate step. Okay. So right between glycolysis, incidentally, where does glycolysis take place? That's right, in the cytosol. Where does the Krebs cycle take place? In the inner mitochondrial chamber, also called the matrix. Your author doesn't call it the matrix, but most other biology books call it the matrix. Um, let's see. So there's a transitional step in respiration between glycolysis and the Krebs cycle. In it, each pyruvic acid molecule that was produced by glycolysis combines with an enzyme called coenzyme A, thus forming acetyl coenzyme A, acetyl-CoA, which enters the Krebs cycle. Now, this intermediate step is important, okay? So, first of all, what's going to happen is you start out with the product of glycolysis, which is pyruvic acid, okay? And that pyruvic acid enters the intermediate step, and there's a geographical change, okay? Which means that it's going to move from the cytosol of the cell. The intermediate step takes place as this thing moves across not one, but two mitochondrial membranes to carry out the Krebs cycle in the mitochondrial matrix. So there's a change in geography, and this is important, okay? So one of the first things that happens is that acetyl-CoA, the enzyme, okay, coenzyme A associates with pyruvate, releasing a carbon dioxide, okay? So remember, pyruvate has three carbons, three. One carbon gets released as carbon dioxide, and two carbons remain behind, and we have two carbons like that. It's called an acetyl group. So this acetyl group gets attached to this coenzyme, and that coenzyme is what finishes harvesting the energy from that acetyl group, okay? So why is this important? Anytime that you see NADH, people, you should think in your mind, high energy electrons. In other words, energy, okay? Anytime that you see carbon dioxide, you should think to yourself, no more energy. All the energy has been harvested from the carbon. Because remember, we've made the argument in the past that the energy in food is in the carbon-carbon bond, okay? So if you broke in that carbon from every bond that it has with every other carbon, you end up with carbon dioxide. And that means that from a biological point of view, that carbon has absolutely no energy left. We have harvested all the energy we can from it, okay? So we end up with an acetyl-CoA, and from that acetyl-CoA, we end up harvesting one NADH. What is that? That's right, high energy electrons. And once those high energy electrons get harvested from that carbon, the carbon dissociates from the pyruvate to yield carbon dioxide. Okay? Now, you have to keep something in mind. There's a lot of accounting. There's a lot of accounting in this chapter, which is okay because you guys are liberal arts. So most of you are probably business majors, so this should be easy for you, okay? So, why does this matter? Because what we're accounting here is for a single pyruvate, okay? But remember, we also want to know what the yield is per sugar, per glucose. And how many pyruvates are made from glucose? Is it one? I'll remind you. Let's go back to glycolysis. How many pyruvates are made? Oops, sorry, that was scary. I've lost me. I can do it. Let's pause. Or maybe I can do it. Let's try to make it bigger and something strange. And it's doing it again. Strange. Anyways, I guess I rolled down instead of up. So we started with one sugar in glycolysis, and because of the glycolytic step, we end up with not one, but two pyruvic acids. Okay? So this is important because throughout this entire chapter, they give you the answer of products based on per cycle, which means one pyruvic acid, or per glucose, which means two pyruvic acids, which means you have to multiply everything by 2. Okay? All right. So in the intermediate step, per pyruvate, we make 1 acetyl-CoA. We harvest high energy electrons in the form of NADH, and we give off 1 carbon dioxide. Okay? But per sugar, you multiply all this by 2. So per sugar, it's 2 acetyl-CoAs, 2 NADHs, and 2 carbon dioxides. Okay? Here's a picture that goes with it. So here's pyruvic acid. Here's 1, 2, 3 carbons. It must traverse not just 1, but 2 mitochondrial membranes. Here, the enzyme coenzyme A gets attached to it. High energy electrons are harvested as NADH, and CO2 is being given off. And the product of this intermediate step is acetyl coenzyme A, which means that we've taken 1 carbon. If you have 3, you take 1 carbon off. 3 minus 1 is 2. So here's the acetyl group, and here's the coenzyme A. So this is acetyl-CoA, and this acetyl-CoA is what enters the Krebs cycle. Okay? So I said, notice that the Krebs cycle takes place in the inner compartment of the mitochondria, or the mitochondrial matrix. Okay? Now, nowhere in the book is it going to say this, and I don't know why, but it takes energy. It takes energy for this pyruvic acid to cross 2 mitochondrial membranes. Okay? You know why? This is a charged molecule, right? Because pyruvic acid, I mean, it's the product of glycolysis. Arguably, it is still a sugar, so it has hydroxides on it, or double bonds with an oxygen. Either way, you have something that is negatively charged. Now, do you remember, can negatively charged Can charged molecules traverse a membrane? Hopefully you remember that the answer is no. The only thing that traverses a membrane is small, non-charged molecules. Relatively large charged molecules don't make it through. So how does this guy make it through? Well, you can do it, but it takes energy. How much energy? 1 ATP per pyruvate. 1 ATP. Okay. And that's where the accountability goes off in the book, because they failed to tell you that 2 ATPs are being used up per sugar, right? Remember, 1 per pyruvic acid or 2 per sugar. So here's the 4-ish step. This author calls it the intermediate step, okay? And it's between glycolysis and the Krebs cycle, and it takes place as pyruvic acid traverses the 2 plasma membranes to enter the mitochondrial compartment or mitochondrial matrix. Okay, Krebs cycle. A lot of neat things happen in the Krebs cycle. Per molecule of glucose is 2 molecules of carbon dioxide, 2 NADH, and 2 acetyl-CoAs. Oh, this is still the intermediate step. My apologies. So in the intermediate step, per molecule of glucose, it's 2 molecules of carbon dioxide, 2 NADH, and 2 acetyl-CoAs. Okay? Now we're going to go to the Krebs cycle, or citric acid cycle. And per cycle, per pyruvate, it's 3 molecules of NADH, 1 molecule of FADH, and 1 molecule of ATP. But per molecule of glucose, remember, we take all that and we multiply it by 2. So per glucose, we make 6 molecules of NADH, 2 molecules of FADH, and 2 molecules of ATP. So here's the entry point. Hopefully it's not a surprise to you that what's entering is acetyl. Hey, I didn't mess it up that time. Acetyl-CoA. Acetyl-CoA. Now, what do we need to know about this? The big points. Here's the big points. 1, 2, 3 NADHs. Why do we care? High energy electrons. 1 FADH. Why do we care? High energy electrons. And 1 ATP. Why do we care? That's what we're trying to make, is ATP. So per pyruvate, which makes 1 acetyl-CoA, we end up harvesting 1, 2, 3 NADHs, 1 FADH, and 1 ATP. Per glucose molecule, what do? That's right, we multiply by 2. So per sugar molecule, we end up with 6 NADHs, 2 FADHs, and 2 ATPs. That's what you need to know. Yay, the electron transport chain. So, yay, words. Okay, we should probably read some of these words. Let's read some of these. Okay, so the movement of electrons through the electron transport chain releases enough energy to power the movement of hydrogen ions. Remember the NADH shuttle? How I told you that was 2 electrons and 2 protons. That those 2 protons were going to have a job. Well, here they are. The movement of hydrogen ions, H+ ions, through the 3 electron transport chain protein complexes, which are also known as hydrogen ion pumps, and they move from the mitochondria's inner compartment to its outermost compartment, which is also called the intermembrane compartment. Why? Because it's between the two membranes. And what is the most important point of this whole thing? The fact that it happens against their gradient. What? This is incredible. So these high energy electrons power proton pumps, and those proton pumps move protons from the mitochondrial matrix out into the intermembrane space against their gradient. Think about that for a second. Think about that for a second. We are accumulating what again? Hydrogen ions. What are hydrogen ions? They're protons. And we are accumulating them against their gradient. In other words, we're producing a proton gradient in between these two membranes. That is incredible. You're not impressed by this? You should be. Because if you're to go out to the most advanced engineer right now, and you tell him or her, I want you to build me a nuclear reactor where we have a bottle of protons, and those protons produce energy. He or she will look at you crazily and go, we can't do that because we don't have the technology to do such a thing. However, your stupid mitochondria that is 3 billion years old can do it. If that doesn't blow your mind, nothing will, because that's exactly what's happening. It is a reactor of subatomic particles. It is accumulating protons, okay? To move a solute from an area of low concentration to an area of high concentration, that's typically enough problem on its own, okay? This is not any ordinary solute. These are protons, which are subatomic particles that have a net positive charge. Do you think that protons that have a net positive charge want to be next to each other? The short answer is Hades, no, right? Absolutely not. Just like phosphate groups don't want to be next to each other because of those net negative charges, protons are very, very tiny, but they have a complete positive charge, and they sure as heck don't want to be next to each other. And this is the kind of gradient that is being built in the intermembrane space of your mitochondria. Wow. Wow. That is a whole lot of power. But that's what you need. That's what you need. You need a whole lot of power because you're producing an incredibly powerful molecule, ATP, adenosine triphosphate. Okay, so against the gradient. So here's the picture. Okay, this is the these folds, which are called invaginations, which increase surface area of the mitochondria, are called cristae. Okay, and here we're taking a cristae and we're blowing it up. So the yellow is the inner compartment, which is called the matrix. And then the orange is the intermembrane compartment that lies between the outer and the inner membrane. Okay? So yellow, inner compartment, orange, intermembrane space. Okay? So now we're going to blow that up, and they're showing the outer compartment, which is the intermembrane space, and they're showing the inner compartment, yellow, which is the matrix. Okay? Here's our NADH. See my NADH? So the NADH is delivering high energy electrons to these things, which are proton pumps. okay? And that proton is being pumped from the matrix to the inner membrane space or outer compartment against their gradient. There's one doing it, and see, we're going to suck up all the energy of that electron until there's absolutely no more energy left. And that electron is going to drive proton pumps that drive protons against their gradient. And when that electron no longer has any energy, it's going to associate with oxygen and two protons, and it's going to form water, and we're going to breathe out water. Okay? Now what? Now we have this beautiful gradient being produced in the intermembrane space, and that gradient is so powerful that it turns this beautiful enzyme called ATP synthase. So as the protons come through that enzyme, it turns it, and it literally mechanically pushes a phosphate on adenosine diphosphate to form ATP. This thing turns like a machine. This is happening in every single one of your mitochondria. If you don't think your body is incredible and elegant and wonderful, contemplate how beautiful this machine is and what an incredible task it's doing as a literally a reactor of subatomic particles, and how the pressure of these subatomic particles is so great that the pressure literally pushes this motor to turn in order to exit. And as they turn this motor to exit, okay, they force an inorganic phosphate to adenosine diphosphate to make adenosine triphosphate. Okay. Incredible. Now, at the risk of making this video longer, I'm going to do it. What you can do this on your own. Let's see. We're going to do a search for ATP synthase. And what's a good one? Here we go. Here's my favorite.
See that bright brain full of activity? That's a healthy 18-year-old brain. Mine is the other one. 55 years old and getting dimmer.
And now I have to wait till the ad. Skip ads.
ATP synthase is a molecular machine that works.
Okay, so first thing that I want you to notice, okay, here's a membrane, okay. These little things, those are protons. Those are hydrogen ions, okay. And this is a very high gradient, so because that gradient is so high, these protons are trying very hard to get out of that intermembrane space. So the only way that they can get out is by going and pushing this door. See the protons here? There's a proton there, a proton there, a proton there. It's kind of like you and I. We're in a motel, and we need to get out, and the door is a revolving door. The only way you and I are going to get out is by pushing that door in a circle in order to get out. Right? So this is going to go in. It's going to make this door turn. And then this is the lollipop head of ATP synthase. Okay. So let's keep on going.
Like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
Hold on. I don't know that you guys can hear this. Let me try this for a second. Ah, there we go. Yay! Volume.
The The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane.
Right? So the analogy of the hotel door holds, right? We're inside, we want to go outside. The only way we're going to get out is we go in, we turn the door, and then we go out. Okay?
In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
So notice that ADP, an inorganic phosphate, associates with this lollipop head. There's 3 catalytic sites on this lollipop head. In other words, 3 ADPs and 3 inorganic phosphates attach. So every time that ATP synthase turns ones, it produces 3 ATPs. There goes ATP.
The F1 ATPase portion of ATP synthase has been crystallized.
When they say crystallize, they mean X-ray crystallization, which means that they know what it looks like molecularly.
Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits.
Do you see the catalytic sites? There's one here, and one here.
It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can...
Okay, I'm going to stop there. They're going to tell you that ATP synthase is an enzyme, so it can work forwards or backwards. Okay, so forwards means that a proton gradient makes ATP. Backwards means that if you have a lot of ATP, this thing will actually spin backwards and produce a proton gradient, which is, again, mind-blowing. Okay, so let's see if I can find my PowerPoint again. Okay, so that's ATP synthase right there. Here's your proton gradient, and here's ADP being turned into ATP as this guy rotates. Okay. Okay, this is important for the accounting. And of course, something's in the way. I don't even know where to put this stuff anymore. Where do I put you? I'm going to put you up there. Okay. It says in the electron transport chain, each NADH molecule will be responsible for the production of 3 molecules of ATP. Each FADH carrier joins the electron transport chain a little later than does NADH, and as such is responsible for roughly 2. So to recap, NADH produces about 3 ATPs, and FADH produces about 2 ATPs. So, and I'm going to tell you, this is a roundabout figure. This can change, and the reason for that is because how much energy is produced is based on the energetics of the mitochondria. Okay, If the proton gradient is very high, the efficiency is higher. So you might produce 3.5 molecules of ATP. If the mitochondria has very little energy and the proton gradient is very low, it might produce 2.5 ATP. So it's not like you can point at it and go, this is exactly what the answer is. It's not true. As a matter of fact, in graduate school, they don't use these numbers. For NADH, they use 3.5, and for FADH, they use 2.5. How do you make 2.5 ATP? You don't. But again, that's a demonstration that how charged or energetic the cell, and therefore the mitochondria is, really dictates how much ATP gets made. But this is close enough for government work. Okay, this is the big picture, people. Okay, here's glycolysis. I put this table in because I couldn't find it anywhere. So glycolysis, how much ATP from glycolysis? Net, hopefully you remember that it's 2. How much NADH? 2. How much FADH? 0. How much carbon dioxide is made? 0. How about the intermediate step? Well, here's the 2 ATPs that nobody accounts for. Remember, we need 1 ATP per pyruvate to traverse 2 mitochondrial membranes. And we have 2 pyruvates per sugar, so that's -2 ATP. Now, how many NADH do we make? We make 2. How much FADH do we make? None. How much CO2 gets released? 2. Leaving an acetyl group behind. Then we go to the Krebs cycle. Per sugar, we make 2 ATP. We make 6 NADH and 2 FAD. Oh, that should be a subscript. I didn't catch that. Can I do that right now? Hello? Hello? Again, let me do it. Oh my goodness. Shame on me. Let's see. Are you gonna let me do it? Font, subscript. Okay. There we go. That wasn't so hard. Now we're gonna go. Now we're in trouble. Now we're in trouble. Now I'm gonna mute myself accidentally. Slideshow from current. Yay! Okay, where was I? Krebs cycle. 2 ATP, 6 NADH, 2 FADH, and 4 CO2s per sugar. This is all per sugar. Totals. Now what I've done in the totals is I've added all these down here. So 2 minus 2 is 0, plus 2 is 2. 2 plus 2 is 4, plus 6 is 10. 0 plus 0 plus 2 is 2. 0 plus 2 plus 4 is 6. Now what am I doing? I'm going to take these guys and multiply them by how many ATPs they made. So remember, NADH makes 3 ATP, and FADH makes about 2. So the 2 ATP just carries over. 10 times 3 is 30. 2 times 2 is 4. And then the 6 just carries over. Now the totals is 2 plus 30 plus 4, which is 36. And that's the correct answer. That's the medical school answer. 36 ATP per sugar. And look at this. 6 CO2s. Remember the equation? Let's go all the way back to the beginning. All the way back to the beginning. Remember the equation? How many CO2s? 6. Why? Because sugar has 6 carbons. So if you have 6 carbons coming in, you have to have 6 carbons going out. This is what's called a balanced chemical equation. And the math shows us that we did a good job. Carbons in, 6 equals carbons out, which is 6. Cool, huh? Okay, now, this is where we need to tell you that this pathway, in particular, Krebs cycle, doesn't only work on sugars, okay? It works on proteins and it works on lipids, okay? So glucose, proteins, and lipids enter the metabolic pathway for ATP production at different points than does glucose, but they enter the metabolic pathway. So in other words, we can harvest energy from sugars, proteins, and fats. Starches and glycogen must first be broken into simple sugars, glucose, before they can enter cellular respiration. Okay, so that's important. Fats must first be broken down into glycerol, which is a sugar, and 3 fatty acids, and these enter the Krebs cycle through what's called beta oxidation, and it makes it makes an acetyl group, which enters the Krebs cycle as acetyl-CoA. Okay, what you need to remember is that fats need to be broken into a sugar and 3 fatty acids before it can enter the Krebs cycle. And then proteins must be broken down into individual amino acids before they can be used for cellular respiration. Now, if it sounds like this is boring, it is not boring, okay? Because all these are catabolic pathways. In other words, we're taking starches, we're taking fats, and we're taking proteins, and we're breaking them down to harvest energy. However, if we need sugar, or we need glycogen, or we need fats, or we need proteins, the Krebs cycle works anabolically to produce these for our bodies. So this is the reason that the Krebs cycle has been hailed as arguably the most important pathway in living organisms, because it is not only where we extract energy from food as we break down food catabolically, it's also where we produce metabolic precursors for, again, glycogen, fats, and proteins. Okay, did I miss one? I always feel like I'm missing one. Glycogen, proteins, and lipids, and this is done anabolically by the Krebs cycle. So it's central. It's as important as it gets. So here's your food, right? And again, proteins must be broken into amino acids. There's 20 different amino acids. So depending on the amino acid, it can enter at different points. It can enter into glycolysis, or it can enter the Krebs cycle at different pathways. If you have carbohydrates, we're going to talk about glycogen or starch. Those have to be broken into simple sugars, and then they can enter the glycolytic pathway. If you have fats, those fats have to be broken into glycerol and fatty acids. This is a sugar, so enters glycolysis. These are fatty acids, so they enter what's called beta oxidation to produce acetyl-CoA, and then enter the Krebs cycle. This, incidentally, is interesting. One of the side products of protein in our bodies is ammonia. Okay, and the reason that this is important is that as we become older, our kidneys have more and more trouble eliminating things that are heavy, and nitrogen is very heavy. So if you're an old person that loves to eat steak in particular, or chicken, or fish, things that are high in protein, okay, then the lower function of the kidneys is going to cause the accumulation of ammonia. Actually, it's called uric acid, but you know, potato, potato. They're pretty close to each other. The accumulation of uric acid in the lower extremities, in particular the feet. And then if enough uric acid accumulates there, you end up with something painful in the big toe that's called gout. Okay. So usually what happens is you have an older person, usually a male, because we're the ones that must eat meat all the time, right? Og eats meat. So we go to the doctor, and the doctor goes, "Well, you have gout. You need to back off on your steaks, and you'll be fine." You know, and then the guy comes home, and the wife goes, "So what did the doctor say?" And he answers, "Well, the doctor tells me I'm going to be having gout every Friday." So, you know, what are you going to do? You can't stop us from eating meat because we're too knuckleheaded. And so we end up suffering from our dietary selection. But the big picture here, I digress. The big picture here is that we can harvest energy from proteins and carbohydrates and fats catabolically, but this system can work in reverse to produce proteins, carbohydrates, and fats. Now this one, not so much, to be honest. Um, we used to have 8 essential amino acids. That has now gone up to 9 because we added tryptophan. We can still make tryptophan, but we suck at it. So what does that mean? There's 20 amino acids, and our body can only produce 20 minus 9, so it can only produce about 11 amino acids. So that means that the Krebs cycle can only produce approximately half of the amino acids. The other 9 amino acids we have to assimilate from the food that we eat because we don't have the enzymes to break them down or produce them, which is strange because stupid little bacteria like E. coli can produce all 20 amino acids, but a big complex animal like us cannot do it. Go figure. Okay, so that's why this guy is so central, because it can either break down food or going backwards, it can produce it, right? So in fact, this is so important because we have to have glycogen in our liver and in our muscles. That the opposite of this is called, the opposite of glycolysis is called gluconeogenesis, producing sugar fresh from a new, gluconeogenesis. So that's something that happens in our liver all the time. And that's it. I hope I haven't bored you guys too much. Um, have a wonderful week. What else can I tell you? And I'll see you soon. I'm going to stop my share. Eating my little guy here. Bye-bye.